feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong, in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong, in my happy place. And I'm right where I belong, in my happy place. And I'm right where I belong, in my happy place.
Hi, and welcome to the National University of Singapore Yong Loo Lin School of Medicine's Healthy Longevity webinar. Thanks for joining us today. I'm coming to you from Riyadh, Saudi Arabia, and my clothes are still in the United States. I guess they wanted to stay on vacation a little longer, so I'm going to be doing it with a sweater today. I hope you can survive that. We have Markus Schosserer with us tonight. It's going to be a very interesting talk about ribosomal RNA modifications, and we'll get back to that in a minute. I want to remind everyone to use the Q&A function to ask questions, and we will get to as many of those as possible. Before we start with Markus, though, we have Max Unfried, who's a research fellow at the Center for Healthy Longevity, and he will talk about a collaborative study on large language models between our lab here at NUS and Georg Fullen's lab in Rostock. The title is Validation Requirements for AI-Based Intervention, Evaluation, and Aging and Longevity Research and Practice.
Hey, good evening, everyone. As mentioned, I'm Max Unfried, a research fellow at the Healthy Longevity Translational Research Program here at NUS. And today I'm going to talk about the collaborative study between the labs of Professor Georg Fullen at the Rostock University Medical Center and Prof. Brian Kennedy's lab here at NUS. And what we do here is we outline how AI, particularly large language models, can aid with analysis of the increasing amounts of data in aging and longevity research. I mean, large language models are everywhere nowadays. Words like Claude, GPT-4, Grok, Llama, have been common in our everyday lives, and many of you probably have tested them out or even used them daily. And what we are interested in is how can we actually use those LLMs for aging biology research, and especially to evaluate neuroscience interventions. If you think about it, aging currently is really going through this era of big data, right, and generates huge amounts of data. And AI, especially LLMs, can assist in helping to analyze this data and improve intervention evaluations. And the idea is that in the future, that better AI-based evaluations can really help us to prioritize which interventions we should be testing in humans, and help us to design studies and even propose like new clinical trial designs. However, to evaluate the results of LLMs, we need certain measures and requirements. And we came up with like a list of 8 such requirements that we should request from the results of an LLM, right? And of course, the results should be correct, they should be useful, comprehensive. It's also important that as a human, we can interpret them and explain them. They probably should mention some causal mechanisms. They should have like a holistic view, also talk about efficacy and toxicity. The results should be reproducible, they should be standardized and harmonized. And especially in aging, it's important that it probably queries like longitudinal large-scale data and also references known mechanisms of aging. And I know this all sounds like a little bit abstract, so let's give an example. And a lot of you probably heard about rapamycin. It's like the longevity scientist's darling because it robustly usually extends lifespan across mammals. But should humans take it, right? And as a human, should you ask an LLM if you should take it? If you were to do that and, for example, query an LLM and ask it, should I take 5 milligrams of rapamycin once a week? And this is me. I'm a 50-year-old male. I'm moderately overweight. I have a slow vaccination response, and my IGF-1 values are high. What it would give you is a response like that. Just it talks about potential benefits of rapamycin. It talks about some side effects, and it mentions that you should consult a physician, which is not bad. However, if we now add the 8 requirements that I just listed, right, that we came up about, that you see here in B again, or if we put into a prompt, here you can have a look at it. If we add that to the query, we actually get improved results, right? In a way, the improvements are added here in green. It still talks about the potential benefits, but it also gives you more context on information that's off-label use, that long-term risks is unknown, and that we actually really need more research in humans to make like better recommendations. However, we find that whenever we add those requirements, the output usually improves, right? And we tested on 2 different popular LLMs. We tested on Claude Opus and we tested on ChatGPT-4. And what we then did is we actually let Opus evaluate the responses of GPT-4 and vice versa. But bottom line is that adding requirements improves the results. So what do I want to take you home, or what I want you to take home with is like that. Well, we came up with 8 requirements for good recommendations by large language models in giving those longevity-related recommendations, and that adding this list of recommendations to the prompt improves the quality of the recommendations made by the large language models. But to be honest, the future of LLM research for neuroscience is just getting started. So keep an eye on it. Back to you.
Thanks, Max. Interesting avenue of research, I think. And we're going to turn to something different tonight, though, and I'm going to introduce Markus Schosserer, who's a principal investigator at the Institute of Medical Genetics at the Medical University of Vienna in Austria. He's done a lot of work on ribosomal RNA modifications, and his title of his talk is Exploring Ribosomal RNA Modifications in Cellular and Organismal Aging. Markus, thanks for joining us.
Thanks a lot for the kind invitation. Yeah, I'm going to talk about ribosomal RNA modifications in the context of aging. And I know that my topic is a little bit of a niche topic in the aging field, but I can hopefully convince you that it's still an important topic. And also, I would like to introduce to you why we actually chose to study RNA modifications in the context of aging. And actually, the basis for that is some preliminary studies from our group in Vienna, but also a lot of high throughput studies done by Brian Kennedy's lab and also Gary Rafkan's lab in C. elegans, for instance, and in yeast, which showed that there's a huge amount of evidence from yeast and from C. elegans that a lot of enzymes which introduce RNA modifications actually are implicated in the regulation of lifespan. So maybe the most studied RNA species in the context of RNA modifications are mRNAs, which you can see here. By far, the most modifications are present on tRNAs, especially here in the anticodon loop. But the topic of, and you see all these enzymes which are indicated in green, these are all enzymes which either extended or shortened the lifespan of yeast and C. elegans, and they introduce specific RNA modifications. But I would like to focus mostly on ribosomal RNA modifications in the context of aging. And as you can see, most RNA modifications are localized in the center of the ribosome, and they are evolutionarily extremely highly conserved. And also these RNA modifications are usually located very close to functional sites of the ribosome. So for instance, they are close to the inter-subunit interface, they are close to the peptidyl transferase center or to the decoding center of the ribosome. All of these RNA modifications on the ribosome, they are installed co-transcriptionally. So when Pol1 transcribes ribosomal DNA into ribosomal RNA, usually very early during this process, even before the ribosomal proteins associate with the pre-ribosomal RNA, these modifications are added. And we also have different types of modifications on ribosomal RNA. So the most common types of ribosomal RNA modifications in humans are pseudouridinulations and ribosmethylations. Of each of those, there are around 100 sites present in humans. Both of them are catalyzed by one specific enzyme. So in the case of pseudouridinulations, it's discarin, and in the case of ribosmethylations, it's fibrillary. And those enzymes which catalyze the modifications are guided to the site which is modified by SNOR-RNAs. So in case that we would like to target specific ribosomal RNA modifications of those types, we need to target specifically the SNOR-RNA. What I would like to focus on in the first part of my talk is base methylations. They are not as common as the other two types, so there are only around 12 sites known on human ribosomal RNAs. And in the contrast to the other types of modifications, these base methylations are catalyzed by one specific enzyme. So for each of those modifications, there's one specific enzyme which introduces this modification. So they mediate both the targeting and also the catalytic activity. And the enzyme my lab is mostly studying is called enzyme 5. Enzyme 5 introduces one specific M5C base modification, and in total there are two M5C modifications known on the human ribosome. One here, which is introduced by enzyme 1, and the other one, which is this one here, which is introduced by enzyme 5. So the modification introduced by enzyme 5 is located very close to the inter-subunit interface, so where the two ribosomal subunits join. There's, in the meanwhile, a lot of evidence that enzyme 5 is highly expressed in several human cancers, and they can also give a good prognosis of the development of those cancers. Almost every week a new paper about this is coming up. And also what is quite interesting is that the sequence which is modified by enzyme 5 is very highly conserved between different species. So there's a stretch of around 100 nucleotides, and those nucleotides, they're completely identical between yeast, C. elegans, humans, and Drosophila. Really indicate, and there are also other RNA modifications located in this area, really indicating that they are evolutionarily highly important. And also in this area, there are only very few ribosomal proteins. So therefore, we guess that really the ribosomal RNA and the correct structure of the ribosomal RNA is really important evolutionary. So we became interested in enzyme 5 almost 10 years, 15 years ago when I did my PhD in Vienna. And at that time, nothing was known at all about enzyme 5. So we've just picked it out from medium throughput screen. And we found that enzyme 5 depletion extended the lifespan of yeast, worms, and flies. Then, of course, we became more interested in that enzyme, and I spent most of my remaining PhD in trying to figure out actually what enzyme 5 does. So we see in C. elegans that depletion of enzyme 5, either by RNAi or also by a complete knockout, so a complete knockout is not lethal, extends the mean lifespan by around 15 to 20%. There doesn't seem to be much effect on the maximum lifespan. The same is true in Drosophila. So in Drosophila, we used two different RNAi constructs, which were also inserted in the genome at two different sites, just to make sure that there are no off-target effects. And also all three constructs extended the mean lifespan of the flies by around 15 to 20%. And last but not least, we tested yeast lifespan. So as most of you might be aware, there are two ways of testing yeast lifespan. There's chronological lifespan and replicative lifespan. So regarding replicative lifespan, we did not see any effect at all. But when we look at chronological lifespan, we again saw an extension of the lifespan. Then, of course, we wanted to know what is the function of ribosomal RNA, of the enzyme 5 modification. So we teamed up with Norbert Polacek in Bern, who is an expert in the structure, in studying the structure of the ribosome. And together we performed DMS probing. This is a technique where you expose ribosomal RNA to DMS, and those sites which are not accessible to DMS, they get not modified, while the sites which are more loose, they can be accessed by DMS and they are modified, and lead then to strand breaks when you do reverse transcription. So we did this in the vicinity of the modification which is introduced by enzyme 5. This is all yeast data, by the way, and we tested different sites which are close to which can be modified by DMS, which are close to the modification site introduced by enzyme 5. And what we observed was quite interesting, because under normal condition, there was no difference at all. So that the ribosome, the structure of the ribosomal RNA was completely the same between wild type and enzyme 5 knockout yeast. However, when we stressed the yeast with H2O2 for 30 minutes, suddenly we saw that the RNA, the ribosomal RNA, became more accessible for the DMS, while after 90 minutes, most likely when the H2O2 is already gone, this effect was again completely reversible. So indicating that this methylation, at least in yeast, might be very important for stabilizing the ribosome, especially during stress. So without stress, there's not much of a difference, but when there's stress, then this modification seems to be important for stabilizing the ribosome. And this actually led us to our conclusion from our first study on enzyme 5, showing that RCM1, this is the yeast homologue of enzyme 5, if we deplete it, we specifically lose this modification. And this then seems to alter the function and the structure of the ribosome. So we see, so we also, for instance, measured the mRNAs which are recruited to the ribosome, and there we saw that enzyme 5 knockout yeast without stress translate similar mRNAs as yeast cells which are usually stressed. So what we think is that the stress response in yeast cells without the modification is constantly upregulated. And this then probably also conferred an increased stress resistance. So we observed in flies that the flies were significantly more resistant to heat stress. Worms were more resistant to paraquat, and in yeast we find both. So we find both that they are both oxidative stress resistant, that they are more oxidative stress resistant, and also more resistant to heat stress. And then, of course, we wanted to know if what we saw in simple model organisms like yeast, worms, and flies, if this is also true in mammals, and therefore we generated a full enzyme 5 knockout mouse. So in our enzyme 5 knockout mouse model, enzyme 5 is deleted in the whole organism right from the embryo stage. So it's not a conditional knockout. We did then a lifespan experiment, and we saw that indeed enzyme 5 also only very slightly extended the lifespan of both male and female mice. The lifespan extension was, as I said, is not very large, and it's also not statistically significant if you correct for multiple comparisons. However, since we are not so much interested in the maximum or in the mean lifespan, but more in healthy longevity, we also measure the clinical frailty index. So those of you who are not familiar with this measurement, in principle, what you do is you non-invasively observe the mice and you look for deficits like, for instance, curvature of the back, graying of the hair loss, vision loss, hearing loss, things like that, which we can observe completely non-invasively. We did this with 3 separate cohorts of mice, and in all 3 cohorts of mice, we saw that enzyme 5 actually was able to reduce the frailty accumulation with age, so the accumulation of deficits with age. This was highly reproducible, as I said, with 3 different cohorts of mice and also highly statistically significant. However, what I probably should mention first, so if you look at the mice and also if you look at the worms and at the flies, you could not see any obvious phenotype on those animals, although they have a complete deletion of enzyme 5. Therefore, we went further and we tried to very, very thoroughly phenotype those mice. And what we found that there are also adverse effects by enzyme 5 knockout. So on the one hand, we see that there's a slight reduction in overall body weight in all age groups, in both in males and in females. We also saw that enzyme 5 seems to be important for embryo development because enzyme 5 knockout animals are slightly embryonic lethal. So when we made 2 heterozygous mice, and we look at the distribution of offspring, we get only 16% homozygous offspring, which indicates there's a slight embryonic lethality. We also looked at the bone density by micro-CT. There we also see, especially in middle-aged males, so in male mice around 12 months of age, we see a loss of reduction in bone density. And maybe the most striking phenotype of all is we analyzed the blood, and there we saw that we see a huge drop in lymphocytes, monocytes, and also in general white blood cells, especially in males, but there's also a trend in females, and this is also present in all age groups, really suggesting that enzyme 5 seems to be important for normal immune function. Now, at the moment, we are still trying to figure out the mechanisms, but we already have some evidence which might explain some of these changes. So we also knocked out enzyme 5 in HeLa cells, and of course, we isolated mouse embryonic fibroblasts from our knockout mice. And in all these in all these cells, we see that the enzyme 5 knockout cells are on one hand slightly smaller, and on the other hand, enzyme 5 knockout leads to reduced cell proliferation. We see this very nicely here in HeLa cells. And we can rescue this cell proliferation phenotype by re-expressing either wild-type enzyme 5 here in HeLa cells or also in MEF. But we cannot rescue this growth phenotype when we express a catalytically dead mutant of enzyme 5, really indicating that most likely this RNA methyltransferase function of enzyme 5 seems to be important for this growth phenotype. We also wanted to know if this association of enzyme 5 with cell proliferation, if we can also see this in whole mice. And unfortunately, there's not a good antibody available for enzyme 5. Therefore, we performed RNA-scope, which is a fish-based method. So we tried to co-localize enzyme 5 mRNA expression with Ki67 as a marker for cell proliferation in kidney and in liver tissues. And we indeed saw that kidney cells which have high levels of Ki67, so which proliferate, they are also high in enzyme 5. While cells which were negative for Ki67, they are also to a much lower extent expressed enzyme 5. Indicating that also in normal healthy tissues, enzyme 5 seems to be associated with cell proliferation. We then, of course, since enzyme 5 methylates ribosomal RNA, we looked at overall translation, and we indeed saw in MEF, so in mouse embryonic fibroblasts, that enzyme 5 knockout leads to a reduction in overall protein synthesis. It's not a dramatic decrease, but it's a significant decrease. And again, we can rescue this deficit by re-expressing wild-type enzyme 5, but not the catalytically dead mutant. Then we also moved forward and tried to measure total protein synthesis in whole mice in different tissues. So we selected kidney, liver, lung, and bone marrow, but here we did not see a reduction in protein synthesis by injecting OPP, indicating that probably this effect of enzyme 5 might be highly tissue specific. So tissues which proliferate very fast, like mouse embryonic fibroblasts, they might require to sustain a high level of protein synthesis, and therefore they might require enzyme 5, while other tissues which are resting and quiescent, like kidney, liver, and lung cells, they might not require enzyme 5 for maintaining the normal levels of protein synthesis. And this might probably also explain why, what I showed you in the beginning, why enzyme 5 is highly expressed in most cancer cells. Just because cancer cells also have a high demand for protein synthesis. We also saw other deficits, so we performed polysome profiling, and we see that in enzyme 5 knockout mouse embryonic fibroblasts, we see an accumulation of the 60S free ribosomal subunits, indicating that enzyme 5 might also be important for the maturation of the ribosome, and especially for the biogenesis of the 60S ribosomal subunit. This is, by the way, something we did not see in other model organisms, so this is something we only see in mice so far. And what we also saw in mouse embryonic fibroblasts is that probably also the quality of translation is affected by enzyme 5 knockout. So we see that in enzyme 5 knockout MEFs, there are higher levels of unfolded proteins, which we also detected by flow cytometry. And last but not least, what we did is we again, like we did before in yeast, we tried to analyze which mRNAs are translated in enzyme 5 knockout and in wild-type mouse cells. And we found that in mouse cells, it was especially mRNAs that are associated with signaling pathways, and especially those signaling pathways associated with development. So for instance, MAP kinase, Wnt signaling, and there were also pathways upregulated upregulated like skeletal system skeletal system development and tissue morphogenesis, which probably might explain why we saw this bone phenotype and also this immune phenotype. Just to sum up this part, so what we think at the moment, we still need a little bit more evidence for proving this hypothesis, but what we think at the moment is that there's a big difference regarding enzyme 5 function between slow-growing cells, like cells resting in tissues, in somatic tissues, which only have a low protein turnover. There actually a slight dampening of protein synthesis might promote aging and delay frailty. Very similar mechanism, probably like with rapamycin or with many ribosomal proteins, which were identified also to extend the lifespan. However, in a more complex organism like mice, there are also some cells which require high levels of protein synthesis for maintaining cell proliferation, like cancer cells, embryonic stem cells, some progenitor cells for immune cells, for instance, and they might really suffer if enzyme 5 is not present, and this might explain the phenotypes we observed regarding the reduced body weight, the reduced bone mass, reduced lymphocyte counts, and also the defect in embryogenesis. So this is at the moment where we are. Any feedback is highly welcome, and we hope for being able to publish this study soon. What my lab is now also trying to do is to identify in more general terms if there might be since we saw these very interesting effects of enzyme 5, we wanted to know if there may be other RNA modifications which might also be associated with aging. And there actually a big limitation until the last 5 years was that there were not really good methods available for mapping those RNA modifications. This was mostly done by next generation sequencing approaches, and there were different methods available. On the one hand, you could induce chemical cleavage of the RNA. There are methods called Ribomet-seq and Hydroxy-seq. You could look at the reverse transcription signature, or you could use antibodies. The big problem with all of those methods were that none of those methods could really analyze all different types of RNA modifications at the same time. So you needed to prepare a library and do Illumina sequencing for each different type of RNA modifications, which of course becomes very costly, and you need a lot of material and a lot of RNA. And another problem was that usually the rate of for false positive hits was extremely high, because especially those antibody-based approaches are problematic because the antibodies are not highly specific. So antibodies which are used for detecting M5C, for instance, they were also known to some extent to react with unmodified Cs, for instance. And therefore, a real game changer was developed and came up and really revolutionized the whole field around 5 years ago. And one of the pioneering labs was Eva Maria Novoa's lab in Barcelona. And she introduced direct RNA nanopore sequencing for measuring RNA modifications. So what we do here is we sequence directly the RNA strand. We have a cDNA strand, but this is only important for stability. What we are sequencing is directly the nascent ribosomal RNA strand. And while the ribosomal RNA passes through the pore, we have a current signal, and this current signal is influenced by the 5 bases which currently pass through the pore. And this is then converted into base called reads. And how do we get now from this raw current signal to some useful information about RNA modifications? At the moment we have 2 different ways available. So on the one hand, we can look directly at the raw current signal. So this is, for instance, an example where we knocked out ribosomal RNA methylase in yeast, and we see that specifically at this position, the raw current signal is affected, while it's not affected in the wild type. So we can just take the raw current signal for getting an idea about the type of the modification of the modified base. The only problem is that also adjacent bases affect the signal, so it's bioinformatically really demanding. But we are very optimistic that in the next few years there will also be specific base callers become available, which really can tell you, okay, now what goes through the pore is exactly an M6A. The method we are at the moment relying mostly on is error-based method. So what we do is we align our base called reads to the reference, and at the sites where modifications are, we get a characteristic error signature, just because the base caller and the aligner doesn't know what to do with these modified sites. So for instance, this site here is modified, and instead of calling a G, for instance, it calls to a certain percentage a C, a C and A and so on. And usually not only the specific base which is modified is affected, but also the surrounding bases. So with this method, this is very good for us for identifying sites which are modified, but it's not yet 100% quantitative. So we can just say, yeah, well, one site changes in this or in that direction, but we cannot say at the moment it's 100% methylated or just 50% methylated. This we can't do. And this is the only major limitation at the moment. So at the moment, we are trying to identify RNA modifications in the context of mouse aging and C. elegans. But I would like to show you, just to finish my presentation, a few slides on our efforts to measure RNA modification in the context of cellular senescence. So most of you might be aware that senescent cells accumulate in our body. They are associated with many different aging-associated diseases. And therefore, at the moment, the whole aging field is looking for xenotherapies, so xenomorphics or xenolytics, which can either make these senescent cells less harmful or completely eliminate the senescent cells. And my lab is mostly interested in skin aging. So we're using we decided to start with two different models of skin aging. So we use primary human keratinocytes, and we expose them either to paraquat, which is a very strong inducer of cellular senescence, or to a mixture of diesel exhaust particles and UVB light to mimic the urban exposome. And then we induced cellular senescence in those keratinocytes. We also, in collaboration with Maria Cavinato and Peter Janssen-Dürr in Innsbruck, we also selected melanocytes, primary human melanocytes, and exposed those cells to UVB light, also to induce cellular senescence. And then we performed nanopore sequencing with the three different senescent models. So our aim was really to see if we can find a unifying signature between different cell types and also different inducers of cellular senescence, because we are well aware that cellular senescence is highly heterogeneous. And we did that, and indeed we find that we identified differentially modified sites on 18 and 28 as ribosomal RNA in all three different conditions. What we also see, so we find that some RNA modifications are indeed uniformly modified. And also we made a very interesting observation, and that is that it always seems that in the senescent cells, there always seem to be higher levels of modifications. So it seems that when cells go into cellular senescence, there seem to be overall more levels of RNA modifications. We don't know really yet why this is the case, and this is definitely also something interesting we would like to follow up. So there are four sites, two on the small and two on the large subunits, which were uniformly changed in all three different senescence models. And especially those two sites on the small subunit are very interesting because they are located in a region, and they were also identified by other labs in the context of different other biological functions not related to aging. So for instance, this specific site here, this uridine, was already shown to be high in different forms of cancer and also during development, and it was also high in our proliferating keratinocytes compared to senescent keratinocytes. And the same is true for an adjacent site, for this G here, which was also high in proliferating keratinocytes and melanocytes, and also high in different cancer models, while it was low in non-malignant tissues and also in our senescent keratinocytes. So it really seems that these RNA modifications, they seem to be specifically associated with aging and senescence, but most likely also with cell proliferation, cancer, and differentiation. So just to sum up, I hope I could convince you that enzyme 5 knockout extends the lifespan of various model organisms and also decreases frailty in mice. However, there are also certain trade-offs. For instance, decreased body weight, partial embryonic lethality, decreased bone volume, and reduced lymphocyte counts, which you have to take in consideration. We think that these different phenotypes might be associated with cell proliferation because we see that especially strongly proliferating cells require high levels of enzyme 5. On the one hand, total protein synthesis might explain these phenotypes, but also specific recruitment of mRNAs into the ribosome. And finally, we used nanopore sequencing, which is highly promising to detect RNA modification in different model organisms. And we already identified a few consistent sites which correlate with cell proliferation, senescence, and development. With this, I would like to thank my group, especially Anja Wagner-Schrittwieser, who performs the nanopore sequencing in our lab, and Lisa Lindl, who did most of the enzyme 5 knockout studies during the Corona time, which was really, really tough. I also would like to thank all of our fantastic collaboration partners all over the world and also our funding sources. And thank you a lot for your attention, and I would be happy to take any questions.
Thanks, Markus. That's a really interesting talk. Lots of questions that I have, and it's already a bunch from the audience too. Maybe just for some people who are non-experts, we could start with talking a little bit about the ribosome. I think that, when the structure of the ribosome was solved, it was particularly surprising, and there were other data to support this too, that the RNAs are doing a lot of the work. People, in translation, people think that RNAs are there just to code for proteins, but they have a lot of enzymatic roles, and I think the ribosome sort of highlighted that. I don't know if you want to comment on that. I just wanted to sort of get that out there for the audience. I think it underscores the importance of these RNA modifications.
Yeah, it's definitely the case. So for instance, we also see during evolution that the bacterial and archaeal ribosomes, they are still much smaller, contain just the core elements, but the ribosomal RNA, and especially those regions which contain modifications, they are more in the core of the ribosome, they're extremely highly evolutionary conserved. And bacteria, they have the very, very similar RNA modifications like eukaryotes, for instance. And then with development and with evolution, during the eukaryotes, the ribosome got larger and larger. For instance, the human ribosome also have this sort of expansion segments where we actually don't really know what these expansion segments are doing. We know that they are maybe important for recruiting ribosomal proteins, external RNAs, but we don't really know the function of them. But for the core ribosomal function and maybe for the structure of the ribosome, the ribosomal RNA and especially also the modifications seem to be important.
Yeah, it's nature's certainly challenging to scientists. It wasn't. We've got not just sequence of RNAs to deal with, but all these modifications that have evolved over time. It's I'm never. I guess I'm not surprised by the complexity just because I've been in the field for so long. It always seems more complex. Yeah, I was going to ask questions about, you mentioned this, that overall protein synthesis is reduced. One of the things we saw in yeast was that, and a lot of the mutations that we found that extended lifespan were 60S. And that led to an induction of GCN4, which I forget the mammalian ortholog of that, but it's a stress-responsive transcription factor. Have you looked at that pathway in any of the organisms? Is it induced?
We did not look specifically at GCN4. We tried to figure out, especially in HeLa cells, which factors might be upstream and downstream of enzyme 5. So we checked, for instance, TOR signaling, insulin signaling, but we did not find any obvious interactions. But there is now some data from other labs showing that enzyme 5 might actually be phosphorylated by cell cycle regulators. And this is probably something which might be quite interesting and to follow up.
Yeah, and some of these cell cycle regulators are linked to aging too, so that might be worth doing. I was going to ask about mTOR. So you don't see changes in mTOR signaling in this knockout, I presume, but I wonder if it could also be downstream of mTOR inhibition. So have you looked at something like rapamycin to see if it affects the activity of the enzyme?
We did not do rapamycin. So what we saw in HeLa cells is that S6 was phosphorylation. There was no impact on that. And we tried also to do some epistasis studies in worms, and there we did not see any additional lifespan. I mean, we still got the by knockout of mTOR, we still got the nice lifespan extension also in the enzyme 5 knockout strain, indicating that there's no direct interaction between them, at least what in regard to lifespan. But of course, many phenotypes actually remind me a little bit of rapamycin. For instance, this very strong immune phenotype that we see in mice, because rapamycin is also known as to have immune suppressive function. I mean, and I know it's a little bit controversial depending on the dose and everything, but somehow it reminds me a little bit of rapamycin, and yeah.
That might be a place to look for interaction in the T cell populations. I was a little surprised by that. Most of the long-lived animal models are have at least at the same or improved immune function. It's these mice are living long. Have you looked at functional parameters of the immune system and challenged the mice with something like influenza or something? That might be really interesting to do to see, because it may be that the immune system is quite efficient, even though there are changes that
No,
you
this is
see.
definitely on our to-do list. The only small evidence that we have at the moment that it seems especially the common lymphoid progenitor cells seem to be affected by enzyme 5. So we see much fewer common lymphoid progenitors, and it also seems that those cells show less protein synthesis. But we did not yet analyze enough animals really to make a strong point about it, but it seems to go in this direction. But we definitely plan to challenge, for instance, mice with vaccines and see if what is the antibody response. And we were also thinking about, for instance, doing bone marrow transplantation and just seeing if the bone marrow transplanted mice still can reconstitute the immune system, even if they're deficient of enzyme 5.
Yeah, I think that's an interesting avenue to head down. I was also wondering about the mouse phenotypes that, this is a standard knockout, right? If you just did the knockout in the adult, you may not have some of the deleterious phenotypes. I guess you may not have the beneficial phenotypes either. So I'm wondering if you're thinking about that experiment.
This I was also absolutely thinking about, and actually our initial idea was to get a conditional enzyme 5 knockout mouse, although we saw that it doesn't seem to be lethal in C. elegans and Drosophila, but somehow the company where we ordered the knockout mouse, they messed it somehow up, and then we ended up with a full knockout, and then we had the option either to wait for another 2 years or do the experiments, and then we decided no, we go straight ahead and do the experiments. But actually, it was planned to do the conditional knockout.
I had a similar thing happen a long time ago when we were trying to make an S6 kinase knockout. So,
Yeah.
we had a lot of explaining to do because at the end of the knockout, there was only one small exon taken out of it, which you would never design either a complete knockout or a conditional knockout that way. But anyway, things don't always go as planned. One of the questions about cancer, it's overexpressed. Is there any evidence that overexpressing it causes cancer phenotypes? Is it just a marker, or does it have a functional role in oncogenesis?
I checked the literature, and if I remember correctly, I think overexpression does, in some instances, it does actually favor cancer progression. I think that there are some studies doing transplantation of cancer tissue to mice, so these xenograft studies. But there is a lot of evidence that overexpression actually promotes also cell proliferation, which would really point very much in the direction that actually enzyme 5 would promote cancer. And therefore,
Yeah, you alluded to the
my personal
stressor.
take home actually is also that if maybe enzyme 5 is because the effect on lifespan and frailty is rather small, but targeting enzyme 5 in the context of cancer might be super beneficial. But still, if we think about that, we still have to think about the immune function, understand better also the adverse effects.
Yeah, it also raises the question of how much of the longevity effect is through changes in tumor onset in the mice. You may not have looked at that yet, but that's an interesting question, I think.
I mean, we did not, because of Corona and not enough personnel, we did not do a full necropsy with all of the mice. But we did some estimations. We checked for tumors which you can feel on the mice, and there we did not see any obvious differences. But of course, it does not replace a full autopsy because mice are very cancer prone, especially on the inner organs, and those of course, you will absolutely miss if you don't do a necropsy.
Well, it may well not be the case too. I mean, with rapamycin, everybody initially thought it was going to be through the reduction in tumors, but it's not. So, yeah, it's hard to know. Maybe I'll ask one more question and then bring Jessica in. So you're looking at all these other modifications now. Do you have indications that some of the other modifications are going to be critical for aging as well, or you may not have looked at that yet. I guess a lot of the enzymes are known to affect aging, so it would make sense that they're having some role. But are there any stories jumping out from the large scale screening yet?
Yeah, so we even before the screen, so we actually followed up one of Gary Rafkan's screens where he identified 2 additional RNA methyltransferases, standalone enzymes, and one of them seems to be really promising because it also seems to interact with SIRT1, for instance, and it really goes up and down depending on the nutrient availability. This is definitely something we are following up at the moment in the lab, especially in C. elegans, which is super interesting. And the other modifications which we identified in cellular senescence, we follow also those up. This is just a little bit more complicated because, as I mentioned in the beginning, those are guided by the SNOR RNA. So there we have to specifically deplete the SNOR RNAs. And this is also a relatively new field. There's not so much methods around, but we now successfully made a keratinocyte cell line where we specifically deleted the SNOR RNA. We really see that the methylation is gone, and now we would like to follow up on that and see possible phenotypes, especially regarding cancer and also not cancer, but regarding wound healing, for instance, cell proliferation, what we can find there.
Cool. Well, I could talk all day. Maybe I'll bring Jessica Liu in, who's here to collate questions from the audience, and we have a bunch of questions. Jessica, what are they thinking?
Yes, thank you so much for the fascinating presentation, Dr. Markus. So one of the most upvoted questions we have is, were there any behavioral or cognitive tests performed for the wild type and ensign NOC5 knockout? I think in mice, I guess.
So we are not experts in that. There are studies actually showing that there are cognitive deficits in ensign 5 knockout mice. So it really seems also to be important for brain development. The only assays we did is we performed open field tests. There we did not find any obvious differences, except that older ensign 5 knockout mice still seem to be a little bit more active and moved a little bit more around. But really regarding the exploratory behavior or going to the center and so on, there was no difference. At least in our hands.
Cool. Okay, I see. And then the next question we have is, does the heterozygous deletion of ensign 5 exhibit the same phenotypic changes as the homozygous deletion?
Regarding lifespan, no. So we don't see any lifespan effect in ensign 5 heterozygous mice. We did most of the assays just in homozygous mice and not in heterozygous mice. And in heterozygous mice, we don't see it. They look much more wild type than they look full knockout. In most of the assays we did, but I have to say most of the assays we just did wild type versus full knockout.
Okay. I have one more question for mice, and then I think we can move on to some molecular ones. The question was, which of the frailty index parameters were improved in the ensign 5 knockout mouse?
This is an excellent question. This is actually something I would now also go back to the data and analyze in more detail. I have the impression it's mostly the condition of the fur and also the vision.
Oh, okay. That's really interesting. So now moving on to a more molecular question. Can the rRNA modification occur on more than one site at the same time?
Can they occur? Yeah, I mean, there are around 220 sites modified in humans, and all of them are introduced co-transcriptionally. We measured all of the modification and actually it seems that of these 220 modifications which are known, it seems that around 200 modifications, or not 200, but maybe 180, 190 seem to be completely essential. So the cell cannot afford to lose those modifications. They really seem to be important for core ribosome functions. While around maybe 30 to 40 sites, they seem to be variable and they seem to change between different conditions.
I see. And to follow up, how stable are these modifications when they occur? So if it occurs in vivo and we extract cells to study in vitro, would the modifications remain to be seen? And yes, so that's part one of the question.
Yeah, so the modifications, they are very stable, and there are no, for ribosomal RNA. So for ribosomal RNA, since they occur very, very early during ribosome biogenesis, you just have to imagine the ribosomal RNA is transcribed, then all the ribosomal proteins attach, and most of these modifications are right in the center of the ribosome. So they cannot really be accessed anymore, and they are very well protected by ribosomal proteins and by the folding of the ribosomal RNA. And also for ribosomal RNA, there are no demethylases known. For mRNA, for M6A, for instance, mRNAs, there are enzymes which are specifically known to remove also the modification. It really seems to be dynamic. But in the case of the ribosomes, it really seems to be associated just with ribosome biogenesis.
Okay.
When the modification is there, it stays there.
I see, I see. And then from this modification, I think I remember you saying that it helps stabilize the stress response, but how long does it last? I guess the question was, is
The
if
stress
that's no.
response.
Yeah, so the question was how long does the stress response with this stabilization effect last?
Leon, in addition to that, how many different kinds of stress responses have you looked at? You mentioned heat resistance and oxidative stress. Are there other forms of stress resistance as well?
So what they look at different model organisms. If we start with yeast, there we did H2O2 stress, 30 minutes and 90 minutes. We did not follow up much longer, but we see also in ribosome profiles that after 90 minutes H2O2, the levels are more or less back to normal. Not fully yet, but I guess after 2 hours, H2O2, the H2O2 will also be completely gone, most likely, and then the levels are back to normal. A collaboration partner of ours also tested high salt concentrations, so osmotic stress. They also see that if the modification is not there, the ribosomes start to fall apart without enzyme 5 modification.
You
That
know,
seems to be
linking that to the mammalian studies, have you looked at induction of senescence knockout? Are the knockout cells more resistant to senescence?
no, we did not do that yet. Because at the moment, the only cells we started were MEFs, and I don't believe that MEFs are the best model for senescence since they are more or less immortalized. But this is definitely also something we would like to follow up in skin. So since we are doing a lot of studies in skin cells, this is also on our to-do list now to make a enzyme 5 knockout in skin cells and then study senescence there.
Awesome. I think we have another question or two more questions for cellular senescence. The first one is, are there differences in the rRNA modifications between cellular senescence and organismal aging?
This we don't know yet. So this is definitely our main question, what we want to answer with nanopore sequencing. So we are now sequencing all the different mouse tissues and also C. elegans. With C. elegans, it was a little bit difficult because there was no nanopore sequencing data available for C. elegans yet. So we first had to establish everything, but this is ongoing. And this is definitely also the aim of our study, really to see if there are sites changing during organism aging, if are there other sites during cellular aging, cellular senescence, and are there some overlaps or not.
I see.
Unfortunately, I can't really give you an answer yet, but this is definitely the work in progress.
Looking forward to the results in the future. And then another question for senescent cells. So you mentioned that enzyme 5 knockout would affect global protein synthesis predominantly in proliferating cells. Now then, in senescent cells, is this something that's possible to observe, and what would happen in that case?
This is actually also some data which we hope to publish soon. So we actually observed that if you compare quiescent and senescent cells, actually surprisingly, senescent cells have significantly more protein synthesis than quiescent cells. So I would expect that if enzyme 5 is not present, probably senescent cells might also run into certain problems because they cannot maintain the high levels of protein synthesis they probably need for producing the SASP factors. But this is also unproven, and this is just a hypothesis.
I see, I see. And then this next question is maybe a little bit future focusing, but the attendee asks, do you think rRNA modifications can potentially become another hallmark of aging?
I would like to, but I would probably not specified on RNA modifications, but in general RNA processing, because I think there's a lot of evidence that by now that RNA modifications seem to influence aging, but also RNA stability and especially also mRNA splicing. So if there would be a hallmark in general focused somehow on RNA, something like, for instance, RNA processing or so, I think this would be definitely considered a hallmark.
Yeah, I think that would make a lot of sense. And when you think about it, the cell spends a huge amount of energy doing this. I mean, if you just look at what's happening in the nucleolus alone, it's a massive amount of energy with the ribosome. So I think we underestimate the role of these pathways.
Mm. And we have evidence. We have hallmarks on DNA. We have hallmarks for proteins, but some of the RNA just in between is missing.
Okay. We'll get there. Pretty soon there'll be 100 hallmarks anyway.
I also think so.
And I guess another question for another future focus question is we saw a lot of results in model organisms and I think some mice and some human studies. And then one attendee was asking, could you share some ideas on how to potentially apply these findings to human beings in the future?
Yeah, as I said, I'm not 100% sure that enzyme 5 is the perfect target for aging. Since the lifespan extension is not huge, especially not in mice, and there's improvement in frailty, but it's also not super significant. But I think enzyme 5 might be a very, very important target in the context of cancer because by now there are so many studies really showing an association between enzyme 5 and cancer. So in this context, it might be interesting. So if I had to develop a drug for enzyme 5, I would go maybe for the cancer indication and not so much for aging. But of course, aging might be a nice off-target effect, so to say. However, but I'm sure that other RNA modification definitely interesting targets. So, for instance, the one which I mentioned before, which seems to be also associated with nutrition, and there are now also a lot of studies on tRNA and mRNA modifications in the context of cancer and in the context of different diseases. And there are even drugs developed by a company called Storm. And probably it would be interesting also at some point to try to reutilize those drugs for aging if there's some evidence that RNA modifications are changed in this context.
I see. So this next question actually asks what could happen if enzyme 5 was inhibited later in life compared to from birth? But maybe we can look at it from the cancer perspective too. And they ask, would this help with any health span therapies?
Yes, so I think if we really think about using enzyme 5 and probably this immune phenotype, since we saw it on the common lymphoid progenitor cells, I think enzyme 5 is really, really important during development. It might be relatively safe to knock it out later in life. This is definitely something would be super interesting to do in mice. I'm not sure if we have the resources for that, but we are aiming to do this in C. elegans using this auxin mediated degon system, where I can very easily deplete an enzyme later in life and then study if this also has some beneficial effects.
So, Jessica, I think we're pretty much out of time. Thanks for doing a great job. And Markus, thanks for being on the show. We look forward to seeing the papers and all the new data on all the new modifications you're working on. So
Thanks a
thanks
lot
for
again for the
joining.
invitation.
And I want to remind the participants to use the chat function and panelists and all attendees option to leave comments and feedback on the show. We'll try to pay attention to that. Our next webinar will be the last of the year, December 19th. Kenneth Meyer, who's a professor and group leader of the Human Movement Sciences Research Unit at Maastricht University. There are several announcements to make before we close. There will be a Healthy Longevity Intensive course for 2025 for healthcare professionals in February 25-26 next year, followed by a Founders Longevity Forum Singapore Unlock Healthy Longevity on the 27th and 28th, which will bring together investors, innovators, entrepreneurs, and researchers in biotech, clinical, and consumer longevity. You can register your interest now. There will also be another Healthy Longevity Talent Incubator, which is 10 days of intense interaction, cutting-edge learning, and unforgettable experiences coming up later in the year. I want to leave you today with a 95-year-old's best life advice for you.
Hey, how old are you?
How old do you think I am?
83.
You're very kind. I'm 95.
And yourself? Almost?
89.
What does it feel like to be 95 and 89?
We don't feel our age at all times. We do at certain times. We each have certain issues, but we're lucky to be walking and out in the sun on a day like today.
If you look at the 20-somethings of today, is there something you don't like about them?
And I think life is more difficult today for younger people than it was when we were young,
In what sense?
in every sense.
I don't think they're as concerned about tomorrow as we were.
We thought of the future.
Well-
I don't think they care. They just spend.
If you could go back in time and give advice to your younger self, what advice would you give to him and her?
Try to be as positive as you can, because tomorrow's going to be a better day unless you make it a worse day.
Is there a cherishable item that either of you have that you've kept with you throughout the years?
One or another. One or another.
I love that.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong, in my happy place. I got my head held high, I'm on